Dr. R.S. Holak, who is Director of School of Public Health, as well as on behalf of our university, Guru Angad Dev Veterinary and Animal Sciences University. So we are very happy you are here, and thanks for, and sorry we are bothering you. You are up early in the morning today, 7 or 6:30 maybe. So thank you very much once again, and over to you, Pikka.
Thank you so much. So very happy to accept this invitation. It's really a pleasure to talk about this topic and in this very important webinar series. So congratulations to the organizers for this, and thank you so much for the invitation. My pleasure. If there's any challenges in seeing the slides or hearing me, then just interrupt me. But I will talk you through different point of views of Toxoplasma, which is indeed one of my favorite parasites. And I hope this talk will make you think about it really from the One Health point of view. So this is really a complex issue, and we really need One Health to solve this or address this, and also to understand this. So I think all that is relevant. So here we have an illustration of the, do you see it as a full screen? I want to confirm once more. Yes. So this is the life cycle of Toxoplasma, and I would say it's brilliant. It's excellent. So this illustration is actually made by one of my PhD candidates, and we will come back to this illustration several times. So indeed, there are many hosts. Mammals and birds can be infected, and key host species is the cat. So cats are important because they are definitive hosts. They are the only ones that, if they get the Toxoplasma infection, they can shed it to the environment. They can make the oocysts. That is the one of the 3 infective forms, the one on the upper corner. Well, in this picture, it's an unsporulated one. This is a photo I took from fresh fecal sample. So this is not dangerous, not infective yet, but after sporulation, which takes 1 to 5 days, then it will have 8 sporozoites in it, and then it is infective to other hosts. And it's also highly resistant in the environment. The picture on the right, the full moon, it's another infective form. So it's actually the tissue cyst of the parasite that has a high number of bradyzoites in it. And this is from a brain of an experimentally infected mouse. And this is the form that is present in the chronic infections in all hosts. And among us humans, about 1/3 of us have this kind of tissue cyst in our tissues, typically brain, muscles, also other places. And here the parasites are slowly dividing and kept under control by the immune defense of the hosts. But this is in a way a game that has been lost because it's staying there. Currently, we don't have any way to get rid of this chronic infection. So it's really a chronic infections living in the hosts. And then there's the third form, the infective form, is the fast and the furious one. So tachyzoids that are in the lower corner picture with Giemsa stain. So this photo is from cell culture. So the really small banana-shaped parasites can be seen. They are some of them inside the cells where they divide, and then there are some already outside. And it forms first like a coffee bean, and then it makes a flower, and then it reaches the stage when it goes to a new cell. And they really leave destructed cells behind in the acute infection. So that is what is happening. It's really breaking the tissue, so this is in cell culture. It's not something I would like to see happening in my tissues. So I'm really happy that in cell culture, there is no immune response to stop it. But usually this acute phase doesn't go on very long. And typically it's causing kind of unspecific clinical signs in most host species, including humans. So there's influenza-like or COVID-like, we would say nowadays, COVID-like symptoms, fever, lymph nodes react, and then the immune system fights back, and the parasites will stage convert to the bradyzoids, to the full moon, and then it's causing much less trouble. So this is really the kind of parasite point of view of the life cycle. In most host species, most of the infections only cause mild clinical signs and then become chronic. But Toxoplasma is really also a pathogen, so it really can cause severe clinical disease that is called toxoplasmosis. And toxoplasmosis, the clinical disease, it can happen after primary infection, but also after reactivation of a chronic infection. And also if a host gets a new infection on top of the one they already have gotten earlier. These photos now, there's two really shocking photos from human cases that are from presentations of colleagues from international Toxoplasma meetings from last year. So there's one showing the classical picture of congenital toxoplasmosis in a child who got infected before being born. I mean, how unfair is that? This can cause a range of clinical outcomes. Also death, but the classical picture is hydrocephalus, calcifications in the brain, and eye lesions. And eyes are another topic really kind of relevant for the disease burden of toxoplasmosis. So another manifestation causing a lot of disease burden is the ocular toxoplasmosis, which is the picture in the center. And this can be a manifestation of congenital toxoplasmosis, or it can be acquired later in life. Other severe forms of toxoplasmosis include encephalitis, inflammation in the brain, especially in immune-compromised individuals, and pneumonia, so lungs can be affected. And those histopathology pictures, they are actually from cats. So domestic cats also, it's not only humans who can have clinical disease. So these illustrations are from cat tissues showing quite severe damage by Toxoplasma. And on the animal side, for example, in sheep, Toxoplasma is a major cause of abortions and also causing, for example, fever and other clinical signs. So really a welfare and health issue, both on the human public health side and in the animal health veterinary side. This is now just a slide showing a print screen of one article, so I'm quite sure you cannot read it all there, but indeed most infections are chronic and subclinical. This is just an example on how our current knowledge already is saying that these chronic infections are not totally harmless. First of all, the infection can reactivate, so we should always think about it that way. Then it has been shown in some animals that this infection changes their behavior. And in humans, Toxoplasma infections have been associated with psychiatric conditions, like here is a discussion about schizophrenia, and also many other things, for example, higher risk of traffic accidents. Challenge is that most of these studies on humans are cross-sectional, so they can only say that Toxoplasma infection is associated with the outcome, but they cannot say what is the cause and effect and so on. But there are a few studies that also support this with having a time element in the study design. Why is this relevant? It is relevant because the chronic infection is indeed very common. As I mentioned, about 1/3 of the participants of this webinar is very likely positive for this parasite and has the chronic infection. Typically, we don't know it. If we have not been tested, we would not know if we have it because the symptoms are so unclear or unspecific. We don't think about Toxoplasma as causing a small flu or influenza-like symptoms. But it's really relevant because it's so common. So this is actually showing seroprevalence in humans in Denmark, where I'm based. So here we have data from more than 100,000 individuals who were tested for antibodies against Toxoplasma from mid-90s to 2010 is the data here. And this is IgG antibodies. So they are long-lasting and very specific. So if a person is positive for these antibodies, it tells their body has reacted against Toxoplasma. And they are long-lasting, so after that's meeting the parasites, the person stays seropositive, we assume, and it looks like that in most cases. And we also assume, and it looks like that then the parasite stays in the body. So it is a chronic infection, and the seropositivity goes with it. What we can see here, there is the age is on the x-axis, and then seroprevalence or proportion positives is on the y-axis. And by during the lifetime here in Denmark, typical life expectancy, about half of the people become positive. But it's not, it goes up with age very nicely, actually. So there's an increase in seroprevalence. So age is very important. And what does this tell? It tells that it's a rather small proportion of all Toxoplasma infections that are congenital, that the people already got before they were born. Most of the infections come later in life. So it is an acquired infection. It's something every day is a risk. When we now today go out there, when you go on your break, I mean, it's there all the time. This just tells that Toxoplasma is around us, and many people get the infection during their lifetime, but not if you take a younger age group there, not everybody has it. So it's also not a thing we should always just, I mean, I would say we can get infected, but we also can avoid it, clearly. Most of the people in the younger age groups have still been managing to avoid it. But it is a high proportion of human population that has this parasite currently. One reason is we haven't been focusing a lot on prevention, I would say. Also, yeah, it's common. But this is why it's important also the chronic infections, the reactivation risk, and the potential links to other diseases and so on, because the disease burden is quite heavy. Here we can also see the red is females and blue is males, and it looks similar in both genders, except they're about the 30-year-old females, their line is lower. This is because this is the data from our diagnostic laboratory, National Reference Lab, and that age group is often when females are tested because they are pregnant and the doctors want to check for the risk of transmission to the unborn child. So they are tested more, and they are tested for other reasons, not because they would have some symptoms that look like toxo. So there's a bit difference there. So the males of that age but more likely are tested if they have some clinical signs that could be toxoplasmosis. But indeed, many live with these parasites, and this tells that we are failing to prevent these infections, or then we are not even trying. And so somehow I feel this is a thing we could do something more, and this is a complex One Health issue, as you will soon, I'm sure I will convince you on that. And this is also relevant because there's no treatment currently that can remove the chronic infections. There are some medications, and new are coming, for mainly focusing on the acute infections. There's been some progress also on the chronic, attacking those full moons, the tissue cysts. But currently we cannot trust on treatment handling this program. And there is no vaccine available for humans either. There is one commercial vaccine for sheep to lower the risk of abortions in sheep, but it's not widely used, and it's not like fully protective. But really what we can do is prevent these infections, and that's the challenge. Prevention is difficult to sell, but we should do it. Why we should do it? In global level, toxoplasmosis causes a high disease burden. That means there is disease, there's suffering, there's lives lost, there is disabilities due to it. And it's been really shown strongly. So I sometimes feel like it should not be a question, this should be a priority. And for example, there's one study from the Netherlands, already a bit older one, where comparing Toxoplasma to other foodborne pathogens, actually how they evaluated the burden, it was causing higher burden than many of those bacterial pathogens we easily think more important, Campylobacter, Salmonella, Listeria. But I would like to say, like, when reading these disease burden studies, be always a bit critical, because how to do that kind of study if it's in these dailies, disability adjusted life years lost, if that's the unit, the system you should, in the study you design all the outcomes of the infection should be counted on. And with Toxoplasma, that's difficult because it causes so many things. So it's a bit difficult to compare to others, but it is one of the methods that allows this kind of comparisons. Incidence is actually a major driver of those results. So if we can lower the incidence, if we can prevent more infections, then the disease burden will also be lower. But indeed, Toxoplasma is priority, top priority foodborne parasites globally, and here in Europe, where I'm based. So this is just a summary of 2 studies that both used a multi-criteria ranking. So WHO FAO study, global study, and a European similar approach study. And in both, Toxoplasma is one of the top ones. There's also some others like Trichinella and Echinococcus and others, but Toxoplasma is globally relevant. That's very important. So back to the life cycle. Now we look at these arrows that you already saw earlier. So really the life cycle of a parasite is the key for how to understand the sources, the transmission ways, and how to prevent. And this may look like it's challenging for Toxo because this figure has so many arrows, but I disagree. I see each of the arrows as an opportunity to do something in a One Health setting, because you can see there's different hosts. Humans are just one of them. And I would say it's not a good idea to leave it to the last arrow. So when we talk about preventing Toxoplasma infections as a public health problem, we easily focus on the humans. But in this figure, human is in the corner. So it's not so relevant for the life cycle. We humans are actually a dead end for these parasites. There is no arrows going from us, except the one, the green one, from the mother to the unborn child. So it is important to note that that green arrow will not happen without the other arrows. So actually the congenital infection, it's only possible if the mother gets an acquired infection. So if either the blue or the red arrow that are going to this human figure here, if those happen, then the next one can happen. So this is like a kind of logical sequence of things. So, oh, sorry, now I jumped, here we are. So indeed, it's important to note that this is like a sequential of the arrows, and we should think backwards. So if we only stop it in that green arrow, there we could do a medical treatment attempt to try to stop the transmission to the child, but that's kind of damage control. So it would be better to do it earlier and also to protect the mother. So yeah, this looks complicated, and I said like humans are in the we are not important actually in this. I would also point out that there's a female figure clearly in this illustration, and preventing Toxoplasma infections in pregnant women is a key priority. But Toxoplasma does not discriminate per gender. So all genders, everybody, all humans can be infected, and we should actually focus on preventing this in all. And here we can really see these arrows, they show the sources and transmission routes, but this looks complicated. And I once commissioned a more simple figure, which doesn't have a human. So this is the one that already showed quickly. So this is based on a sketch I made on a piece of paper. So this is the life cycle Toxoplasma seems to be very well adapted to. There's the cat catching the small animal, getting infected by carnivorism, eating meat or tissues. And the cat is the only host that can leave the Toxoplasma oocyst behind in its feces. And then there comes the small animal and gets that from the fecal oral route, and so on. So this goes kind of round and round. And actually, this could be without the cat. So if the little animal shown here would be eating tissues of another animal, only the carnivorism, that's enough. There's no need for the oocysts and the cats. But the cat is very beneficial for Toxoplasma because it amplifies these parasites. A cat that gets an infection, it can get it from a single produce, so it's single little parasite in that full moon picture I showed earlier, and then it can shed millions of oocysts. So it's really amplification. The shedding is short term, but it can be repeated, but it's really efficient. And why cats? Actually, the explanation for this is known now, since quite recently. This was published last year. So all cats, all felines, lack a specific activity that is needed for their metabolism, and this metabolic inability actually gives them the superpower of being definitive host of Toxoplasma. So cats are the only ones that can naturally provide what the parasite needs for its sexual reproduction, and that is why the cats are the only ones that can shed the oocyst of the parasites in natural infection. And other hosts, including us humans, we cannot do that. We can only support Toxoplasma by getting infected and allowing it to grow in our tissues, and then somebody should eat us, and then it can go forward. There are also other ways, like the transplacental infection, vertical directly from the mother to the offspring or fetus, and then also infection from transplants is possible. And in some host species, Toxoplasma has been shown to be transmitted in milk and in semen. But the main routes are eating it in different ways. So back to the arrows. There is a color coding here. So it shows the three main ways. Carnivorism is the one in blue. Fecal oral is the red, there's the oocysts, and transplacental is the green. So the cats is, yeah, there could be actually few more arrows added here, but this shows the main routes. And for example, the farm animals, they are assumed to be being herbivores and eating mainly plant material. So there is no blue arrow to them. But for example, it's known that some like pigs they are omnivores and they might be catching rodents and so on. So this is well known from the Trichinella field, for example. So there are also other things, but this shows the main things. And there is no blue arrow from the cat to humans, because in our region, cats are not usually consumed by humans. Cat meat is not eaten by humans, but there could be even more arrows. Key point of my talk is that there are two arrows coming to the human. The red arrow, well, we don't eat cat feces or these oocysts directly, but actually they need to be incubated the 1 to 5 days before becoming infectious, and it's not directly cat feces, it's something contaminated with cat feces. And also there's been food outbreaks, but also several waterborne outbreaks, so that's quite typical. And as I mentioned quickly earlier, those oocysts are highly resistant in the environment, so the cat can be long gone and the oocysts are somewhere in the environment or water, and humans can get infection from that. In Europe, it has been concluded based on quite few studies that the main transmission route to humans is the eating meat of infected animals, and needs to be eaten raw or uncooked. And it's actually interesting that our meat inspection, while it's focusing on some parasites like Trichinella, meat inspection currently does not even try to find if this animal that we are planning to consume as human food, if this animal is infected with Toxoplasma or not. So it's kind of allowed to be there. So the raw meat that consumes eats and all the medium steaks served in restaurants, they can have this parasite. It's allowed, and it's microscopic, so it's not visible. Can be there, and that's why it's kind of interesting. Of course, cooking will kill it, so that will do it. And one thing is also that source attribution, finding out what is the source, it's quite challenging for Toxoplasma, and this has made it difficult to target any preventive measures and interventions. So overall, we really have a question that should be answered, and that is where this infection actually comes from. So here is actually another illustration of the life cycle from a recent EFSA, European Food Safety Authority document, really showing the same thing, that there are several transmission routes. Here are, for example, bivalves also shown. And where should we focus our efforts? Where should we go? Is it which of the arrows we should focus on? And it is really a question. So putting the question different way, what is the relative importance of the foodborne pathways? For example, food is quite easy as a focus where we could do something. Because if it is really the meat the most important, then we should focus there, or is it the oocysts? So currently we kind of lack this information, and I'm super happy and proud to be, as Reinish mentioned, I'm a project leader in a large European project called Toxoplasma. So we received last year 3 million euro funding as a One Health consortium to look into this, to look into the sources of Toxoplasma infections. And there's a website, easy to find, also just Googling Toxosources. So we have more than 20 partners across Europe, institutes that are focusing on public health, others animal health, some on food. So it's really a One Health consortium. And it's so clear that for Toxo we need this, and I'm super proud and happy about this. It's only 2.5-year project, so we are pushing for it, and we will also, there will be results coming very quickly, so stay tuned. But it would not be possible without this kind of collaboration. And I must say, this year with the COVID-19 challenges and all the lockdowns and everything, it's been also a strength to have a consortium, because then different places people can work, and we've been able to progress. And in that EFSA study, actually, our background, where do we start when we would like to, in our Toxosources, to look into the different sources? So what was known before? There is this, let me see, there's chat comments coming. Can I see it? I'll just email so we can see, ma'am. Thank you. Perfect, sorry. I need to close that. So, what was known beforehand? So 2018, EFSA, the European Food Safety Agency, they published a scientific opinion on this topic. And it was a discussion, how can we know? What tools do we have? And first of all, there are epidemiological studies, of course, that could answer this question. Case control studies, there are a few. Outbreak studies. There's not so many outbreaks or outbreak investigations published or done on Toxoplasma, some on waterborne, very few I will show in a moment on foodborne. But the challenge there is perhaps the unspecific clinical signs, so maybe they go unnoticed. There might be more outbreaks of Toxoplasma, but we don't notice them. Then there is a study type comparative risk assessment. So this QMRA is quantitative microbiological risk assessment. It shows if you have different types, and you can say which one of these is most important. For Toxoplasma, it has been focusing on meat-borne routes, so different meat types. Which of the meat types available in a country is most important source? That has been kind of the question. Then also there's been attempts to find new methods. So methods that when we diagnose the infection, we could say if this comes from this source or that source. And there's some publications, some attempts and approaches, but not too many. Then, let me see if you go forward. There you go. Then there's, of course, more genetic molecular approaches. So subtyping, Toxoplasma gondii is just one species in the genus, but it has several genotypes. And that would be, of course, a way to, for example, investigate an outbreak and tell what is the source. The challenge is that the current methods, they lack the discriminatory power to really see different types. At least in our region in Europe, we have mainly type 2. So it doesn't help so much, because almost all we find is type 2. If I have an outbreak and I have a potential source and some cases, type 2 is what I expect, and then it's difficult to say if that's they are linked or not. And then there's been use of expert opinions or expert elicitations. So asking experts, based on what you know on Toxoplasma, where do you think the infections come from? This is, of course, not really data-driven. This is opinions based on knowledge, but it's still not so high-level evidence. So concluding remarks in this documents about how to do source attribution for Toxoplasma was that it seems to be a major thing that foodborne, it is often foodborne, so food sources could be one focus to focus on. I would like to mention that this was European Food Safety Agency's scientific opinion, so of course their focus already is food. Major sources, we think it's meats and vegetables, so it is foodborne, and then water is also known. And there has been this evaluation of what type of meats could be the most important. It would be really excellent to have some methods that would be telling, especially serology, like I showed those 100,000 serology results. If we could use a method to say this proportion came from oocysts, this proportion came from the tissue cysts, that would guide us a lot to see where to focus. And indeed the typing, that would be, it might become better because the progress in that field is so quick. So here are actually 4 slides that were presented by Dr. Marieke Opsteeg, who is one of the persons in my consortium. These were presented in the One Health EJP's annual scientific meeting earlier this year. So this is an overview of 9 outbreak or case reports from Europe. Most are individual patients or a household, and it's really based on reported exposure, and there was not really direct detection of the parasite in the potential source. So it's quite limited information. There's a few other studies from other parts of the world, but very often same challenges, like how strong is the evidence. This is then about risk factor analysis that have been found. They do provide a better way to associate the exposure to a potential source with the probability of the infections. And there has been various risk factors that have been identified and looked into. Usually there's an odds ratio, sometimes risk ratio. It shows the information how the probability of the infection increases if there is a certain exposure. So if you eat this, then you have this much higher odds to be infected. But these are also not too many studies. And then there's only 2 with the population attributable factor. That's in a population level, what part is the likely source. These 2 points out to undercooked meats, for example. But there are also clearly showing that there's different types of consumption and habits, so many things. So the Cook study is well known and highly cited. That's very well known. Expert elicitation, there's two on this summarized here. So food is considered more important than soil. So this supports the thing that EFSA was focusing on food too. And but sometimes these categorizations are not so good because food is many things. And I mean, there is for Toxoplasma, for example, the meat and the vegetable route or the water. So it depends also sometimes when we have a system from other pathogens, it might not work so well for Toxoplasma. But then there have been these quantitative risk assessments done on relative contributions of different meats. So these have been only meats. So this is I think a major limitation. So there's one that, for example, showed looked only one type of meat and then like showing that which types are most important. But then there's 2 studies in Europe, one in Netherlands, one in Italy, both suggesting that beef, meat from cattle, would be more relevant than pork, meat from pigs. So even though the prevalences are the opposite, but it's about how it's eaten. So it's in these models, it's very important how the meat is prepared. And then also, what is the prevalence of the pathogen in the meat type. But I would say not so many studies here either. So indeed, the EFSA opinion concluded that the main gap is that the relative importance of the main transmission routes from the environment and from the meat. It's a major issue, and we currently can only base it on experts' opinions based on these few studies. So there is really a gap. And that's something where in our toxosources we are working on. We are looking what are the relative contributions of the different sources of toxo infections. And our key objectives are making these kind of quantitative comparable estimates, but we will include also the environmental, the oocyst routes. So not only meats, but also the vegetables and others. And we have some specific aims, like ready-to-eat salads are a potential source. And we are looking into, let me see. Oh, there was just mine is fighting. So that is one kind of specific gap. We want to look into the salads, because eating salads that are actually sold so that they are ready to eat is gaining popularity, and those could be also contaminated with Toxoplasma, and it has not been studied so much. And then we also focus on method development, exactly addressing these gaps that were shown. So both for serology and then for the typing method. So this is we are already working very hard on this, and I look forward to report results very soon. And let me jump. Whoops. This jumps here. So until we have more information about the relative contribution of those roads, so that's those arrows going to the human, the red one and the blue one. So, and probably also after that, we do need to focus on both arrows. So we should stop these infections better. While I'm talking, I would like you to also think, what would you do? So some of the things are very obvious. So if we would not eat raw meats, that is very clear way. Indeed, if it's heated to 67 degrees, the parasites will be dead and not infectious. I would say my personal respect for Toxoplasma is one of the reasons I eat very little meat, and it's never undercooked. So it's I acknowledge this risk. Milk, if it's pasteurized, same thing, then there is no risk. Washing vegetables that are eaten without cooking, it's important. Using gloves when doing gardening and work with the soil, and eating clean, drinking clean water, keeping water clean is important. People who have cats, they should empty the cat litter box every day, because the parasites are not yet infective when they are really fresh. So that would be a clean way to get rid of them. And if we have cats running around, they should not be allowed to defecate in places that could contaminate something we eat. But I mean, as the infection is so common in humans, it seems we are failing this point. And as I mentioned earlier, this is actually the last line of defense. This is the last arrow. So in a One Health setting, we really could do more. So now we go to the One Health thinking. We go upstream the arrows, and remembering that each arrow only happens if the previous arrow happened. So it is not actually undercooked meat that is a risk. It is muscle tissue, that's not a problem. But it is undercooked meat of infected animals. So it is muscle cells that have infective parasites. So then we come to the question, how common is this in animals? And it is very common in animals worldwide, in animals that we humans eat. This paper we published last year from our region estimated pooled seroprevalence, again using serology for toxo. It was 6% in domestic pigs, 23% in sheep in our region. That's very high. And indeed, also, we should remember that this is also a clinical disease in animals. So not only the source to humans, but also as an animal disease, we should do something here too. So how do these animals get their infections? Could we prevent that? Of course. The way we have these animals now and raise them for their meat, they are kept, I mean, we humans are controlling them. So they are mainly herbivores, and they mainly get plant based food or feed. So that they get the infection, they are eating or drinking something that has those oocysts. So it's the red arrow going to the cow's mouth here. So that we should stop. And as we can see, high proportion infected, it tells that this fecal oral route is well established. In our food production, the animals we are raising for food, they are actually eating something that has fecal contamination. That's not really okay. So first of all, this fecal oral transmission should be stopped. There are also other pathogens that transmit this way, so that would support it. Feed hygiene, clean water to animals, pasture hygiene, making sure that the cat feces don't contaminate what our farm animals are eating. And going upstream from there, those red arrows, they go to the cats. So important there is to see that they don't come from the cat if there's no arrow going to the cat first. So again, we need to remember that it's not just cat feces that are a risk. It is feces of a cat that had the infection and is shedding those oocysts. Is that common? Yes, it is, just like in humans. Also in cats, the seroprevalence increases with age. Here are studies from Finland and Estonia from my region. And with the age of the cat shown on x-axis and proportion positive on y-axis, very similar to the humans. So it's common, and cats also get it. They also acquire it during their life. It's smaller proportion there in the young ones. And what is important to think about these figures, it's about 50% of cats, 40-50% of cats that are positive. And all those positive cats have been doing their superpower for toxo and have been very likely shedding the oocysts in their feces for the 1 to 2 weeks short time. Then it's up to us humans who are taking care of the cats. Where did those cat feces end up? So if they were disposed correctly, landfills, burned, somehow contained, that's not a problem. But if that cat was running around and the cat feces are contaminating our gardens and food production, then it was causing a problem. So then they are out there. So it's really tells that way. And as I mentioned, it really is also a disease in animals and also in cats. So most of those infections, those positive cats, they don't have any clear clinical signs of the infection. But in cats, generalized toxoplasmosis can be even fatal, kill the cat. So this is a study I published from Finland, and there we had in the veterinary pathology units, the cats that came there for necropsy. 3% of them had died from toxoplasmosis. That's not a very small proportion, because there's many reasons for death for sure. And Finland overall, there's very few infectious diseases in cats because cats are vaccinated and usually like pet cats taken care of. So it is actually relatively common cause of death even in cats. Important here is that indeed that there is no arrow coming from the cats if there is first no arrow going to the cats. Only infected cats can shed the parasites. How to prevent the infections in cats? Well, there are the arrows going to them. So very clearly, no, if we would not allow the cats to hunt, which they do, of course, naturally, but if we would keep them so that they're not hunting infected rodents and other animals, that would be a thing. But another important thing, at least in our studies, we've seen for pet cats, if the owner is giving raw meat to the cats, that is a major risk factor. So raw meats is not only to humans, but also to cats, a relevant thing. And one key thing also here is what we could do is getting rid of the cat feces properly. They should not end up to our farm animals. They should not end up to our salad, our drinking water. So that is really important thing. So I think also seeing these arrows where we can stop it, this is really a excellent idea for in this webinar to discuss and think about how One Health is important to solve a complex issue. There's many routes, many things happening, many arrows. In zoonosis like toxoplasmosis, we cannot solve it with only looking at humans or only looking at single animal species. So it's really needed to have the One Health. Thank you for your attention.
Thank you very much, Pikka, for such a wonderful talk. So I hope all the participants have enjoyed it. And now if there are any questions, you can write down those questions in the chat box. So there is one question already we have. So it is, what is your opinion? Do you want me to read, or you can?
I can see it. Yes, so I can mention it's just opinion on use of animal feces as manure, or actively putting it into the. That's actually something that for toxo, because we would not typically use cat feces for that, so it's not really relevant for toxo. But there needs to be understood the risk of actively spreading fecal material. What pathogens could there be? And so that is possible thing, but for toxo it's not so relevant because cat feces are not in that amounts, and cats are the only hosts that can have this parasite in the feces. So that's not relevant for toxo, but I think that is very important point too. It's a very useful material for fertilizing, but we need to always remember the pathogens there. And then is poultry safe from this infection? No. So indeed, Toxoplasma can infect all warm-blooded animals, so mammals and birds. And then also it can kind of be contaminating or present also in bivalves, in mollusks and so on. But if poultry the poultry production is very intensive, then usually it's possible to keep the parasite out. But free-ranging chicken has been used as an indicator host for Toxoplasma in the regions, and for seeing which genotypes they have. So they are picking it up. So it is a possible source. But again, only if it's raw. So well-cooked meat is not a risk. In general, for diseases spread via fecal oral route of transmission, I would just say that is actually, for toxo, I feel like we've forgotten that. We talk about the meats, and then when we actually put the name to it, that it's a fecal oral, then we realize that there's this basic hygiene things that we could still do so much better. So and really good point that it's for many diseases. So it's actually a point to focus on that way. So that's really the drug of choice for treating toxoplasmosis. That's actually, there's what is available and what is done so far. There's discussion, for example, for congenital toxoplasmosis. There's not really a good answer. So basically sulfa type drugs with pyrimethamine are used routinely, and then there are also spiramycin treatment protocols and so on. And there are some new ones coming too. And in animal side, clindamycin is also used. But actually, that's something that is really discussed. Last year, there was a congress on congenital toxoplasmosis in Italy, and there seems to be changes in this, but also there is not. It's a bit challenging to do the studies what works, and there is usually also the link to the diagnostics, when to treat and what to do. But then there's also like the immune compromised, so the current routine treatments usually keep this away, so then there's not so much need to treat also. So we usually try to be ahead of time. That's a key thing. Whether cats get lifetime immunity after infection, really important question, because we always previously said that cats shed only once in their lifetime. But there's been a few studies. Now, I don't remember the authors, in Veterinary Parasitology published a few years ago, where they challenge cats again with a bit different type of parasites, and they were shedding again. Then there has been also some studies where if the cat's immune response level goes lower, if they become immune compromised, then they might shed again. So it's not immunity, not total immunity. It seems to be actually that in many hosts. Also in humans, we used to think that for pregnant women, for example, that if you have been infected, you are immune. But there are several case reports of a new infection that has then caused congenital toxoplasmosis in a child. So it's not 100% protection, but it is really immunogenic. It's really causing a reaction. So something happening. Then they say a specific breed of poultry. Actually, breeds are really interesting. I have myself been doing a study on cat breeds, and we could see differences in their Toxoplasma prevalence. Also, when we took into account their different lifestyles, do they get raw meat? Do they go outside to hunt? So there could be something in the host genetics. But for poultry breeds, I haven't actually seen. I don't remember seeing studies. If you know, let me know. I would be happy to read. But in sheep also, there's a few studies about breed differences. So that could be a relevant thing. Any vaccine trials against toxo in cats? Excellent question again. Very good questions here. There has been actually also published some like kind of research use vaccines for toxo for cats. The challenge is that they are quite far from being able to be used like more widely because I think it was a live vaccine and you had to vaccinate every 6 months. So it's quite challenging to get the vaccine coverage high enough, because it's been modeled that it should be very high vaccine coverage for it to help. But it could be a good addition to the toolbox. Personally, I think we could focus on stopping the cats from getting the infection. When I saw our own study results that the raw meat given by the owners was a major risk factor. I mean, if the owners would cook the meat they give to their pet cats, that would help. And also thinking about how we keep cats. We use them for rodent control, but that is quite old-fashioned way in a way. I think in 2020 we should be able to use other means for that. Then there is, if a carcass is positive for Toxoplasma, can other animals be slaughtered after treatment, or the whole herd has to be culled? No, actually Toxoplasma is indeed allowed. It's okay. I mean, nobody's taking it as a problem that 20% of our sheep are positive for Toxoplasma. So it is not, if a specific carcass would be found positive, first of all, they are not tested now, but it would be just going to a normal consumption. Currently, there is no limitation of that. It should be cooked. That's the point, and there is no effect to the farm or the other herd. So no major reactions. And then the same for milk production. So milk actually as a source is not so much studied, but no, currently no. And I think that would be perhaps too drastic to think about cooling all the animals, because the prevalence is so high. So we need to start it a different way. Then you said acute infection can cause vertical infection. Why not chronic? Well, the problem is that actually from the chronic, if it activates, then there will be again the active parasites, the fast and the furious going around. The chronic is usually so well contained that they say that if a woman has been, for example, infected and has the chronic infection, usually it doesn't pass it to the child. But if the woman's immune status goes so low that those full moons, if the parasites activate, then they can go again. So kind of the sleeping dormant chronic subclinical infection should be kept under control and not causing problems, but it can. So reactivation is possible. But most of those, the focus is in pregnant women to prevent the first infections, the acute infections, because that seems to be the main thing, that if there's a primary infections just before the pregnancy or during the pregnancy, then there is a risk for transmission, because the parasite is so actively all over the body.
I think there are no more questions.
Excellent questions. I'm so happy, and I hope you will everywhere continue discussions also during your break, because this is a topic that comes close to everybody. So it's really it is a One Health topic.
Yeah. Okay, there is one last question. I think we will take this last question.
Yep.
I once read somewhere that toxoplasmosis can be attributed to high crime rates in some countries such as Brazil. So would you?
I mean, this is exactly what I said, that these chronic infections have been associated with many things, also behavioral things, or like car accidents, for example, there's one study. Challenge is that which comes first. So sometimes, often these studies are, you just say they are positive for Toxoplasma by IgG antibodies. You don't know if those antibodies are, I mean, which came first, the crime behavior or the parasites, but associations have been shown. This study I don't remember seeing. I need to check that. Sounds yeah, interesting.
Okay. Thanks, Pikka, and thank you very much for answering all those questions. And I highly appreciate all the participants who have really involved actively or throughout this talk, and then they ask questions. And I highly encourage students. We do have undergrad students, Pikka, in this webinar. So as well as our master's, PhD students, as well as faculty all over from India is attending this webinar. So we are very happy that more than 200 participants are, and this is one of the highest number we are having in this webinar. So thank you very much for your wonderful and interesting talk. And at the end, I would like to thank, first of all, our Vice Chancellor, Dr. Inderjeet Singh, who has always supported us to or organize such webinars. And then Dr. Ghuman, who is Dean of our vet school, as well as Dr. Olak, who is Director of School of Public Health. Once again, I would like to thank all the participants. Thank you very much for registering as well as participating in this webinar. And I hope that you will continue involving or attending these webinars in the coming weeks. Thank you very much, all the participants. And before we end, I once again would like to thank Dr. Pikka on behalf of our institute. Thank you very much for your time, your efforts you put in your talk. We are really happy. We are glad that you have spared your time to give this talk. Thank you very much, Dr. Pikka. And I want all of you to join me in thanking Dr. Pikka.
Thank you. My pleasure. It was really wonderful. So yeah, all the best to everybody. We'll see.
Thanks, Peter. Bye now.
Thank you. Bye-bye. Thank you.
And then I ask participants to please fill that form before leaving, which has been posted by one of our colleague or team member. So please fill that form, and I hope that it will be another 5 or 10 minutes before, let's say another 5 minutes, we can before ending this meeting. We have another 5 minutes. The link will expire 15 minutes after webinar. So you have 15 minutes to fill those Google form, which will be required to, for your attendance, right? So another 15 minutes before we end this meeting. And once again, thank you very much to all of you. Thanks.